Good morning. As Lisa said, my name is Katherine Beaverson, and I've recently joined the Dyne organization as Vice President of Global Patient Advocacy. I just want to thank Defeat Duchenne for this amazing opportunity and amazing conference today. And certainly many thanks to all of you who are in the room and online to hear these presentations. I'm a little intimidated. The previous speakers were excellent. And my good friend Paolo is coming next, so I'm kind of in the middle. Okay. Next. Okay. Just want to remind everybody that Dyne Therapeutics is a public company, and we will be making forward-looking statements today. You can refer to our website for more information. So our why is really stated in our mission, and that mission is to deliver life-transforming therapies for people living with serious muscle diseases. And this mission is woven deeply into our core values and inspires our employees, our dynamos, each and every day. So I think as previous speakers had referenced, right, to deliver life-transforming therapies, we really first have to understand what's the unmet need experienced by individuals and families with Duchenne, and collectively, and I think with all these companies in the room, it's true collective effort to seek to solve for the challenges faced by these families. And when we specifically think about approaches to restore dystrophin, um, in individuals with Duchenne with the goal of slowing progression of disease or perhaps even improving function, there's general agreement that the current PMOs have significant limitations on dystrophin restoration and functional improvement. And three specific challenges contribute to these limitations. And I think, Elena had referred to them as well as Drew. And that really is, can we get oligonucleotides into the nucleus to really harness the cell's natural machinery? We are limited in targeted delivery specifically to muscle fibers. And the other challenge currently with existing oligonucleotides is that broad distribution to and in the muscles that matter the most in Duchenne. And so our solution, we believe the solution to overcoming these challenges rests with the promise of the FORCE platform. And the development of Dyne 251 as a potential next generation exon skipping drug that can produce high levels of dystrophin to provide true benefit in function and in quality of life. And I'll just remind folks online and in the room and more broadly that this is really a part of an ongoing conversation with the Duchenne community about the FORCE platform approach, its trajectory through preclinical research, and now a demonstration of that platform approach translating into the clinic, both from a safety and tolerability, as well as efficacy perspective in our DELIVER trial of Dyne 251 for Duchenne. A reminder that the FORCE platform consists of three components, one being the antibody fragment that targets specifically receptors that are located on the cell's surface, widely available on muscle cell surface, the linker, which is that portion that joins the antibody fragment to the PMO, and then of course the PMO, which is that portion of Dyne 251, which performs the precise exon skipping. Now, more about that receptor. We deliberately chose to target the transferrin receptor 1 or TFR1 because it's highly expressed on muscle cells, those that matter for Duchenne's skeletal, respiratory, and cardiac. And its natural function is to help move iron into muscle cells. So by design, Dyne 251 is meant to take advantage of that natural process of cell function by using the TFR1 receptor to deliver the exon skipping PMO to the muscles. affected by Duchenne. So what are we doing, right? Dyne is currently conducting the DELIVER trial, which is a global phase 1/2 clinical trial, again, to evaluate Dyne 251 in patients with Duchenne. Detailed information can be found about the study on links such as clinicaltrials.gov as well as euclinicaltrials.eu. And you can see that there are 3 participating sites here in Canada with Dr. Hugh McMillan yesterday, with Dr. Craig Campbell, as well as Katherine Selby. I will remind, because I think we've made this a key part of our conversations previously, that prior to starting our Dyne 251 trial, we proactively obtained expert feedback from patients, caregivers, and advocacy leaders, such as those affiliated with Defeat Duchenne. And they've provided insights into, based on their experience of living with Duchenne, their voice of the community, as well as references to past and current trials. And this input has been critical to ensuring a patient-centric design that has included things like key considerations for choosing a clinical trial. Why would you choose one over the other? What are the services that can be provided? Review and input on our informed consent documents. These are overwhelming documents. How do we think about the language? How do we think about education in terms of key concepts that allow true informed decision-making? Feedback on elements such as biopsies and functional outcomes. What are the ones that matter the most, and how many biopsies will folks tolerate? Identifying the most burdensome aspects of a clinical trial and how to ensure that folks are supported during their experience. And I think we've heard powerful descriptions of their experience by Terrence, by Torrance, by Eric, and by Elijah yesterday, so everyone can appreciate how those insights and experience really do contribute to the way we think about operationalizing and feasibility elements of a clinical trial. So the DELIVER trial is enrolling ambulant and non-ambulant males with Duchenne who are ages 4 through 16 and have obviously mutations amenable to exon 51 skipping therapy. We do include patients who've previously been treated with exon skipping therapy. They can enroll following a 12-week washout period. So I think that addresses, a previous question online when we think about other therapies and how we incorporate them into the elements of a study design. The primary endpoints, as Dr. McMillan went through the trial process yesterday for phase 1/2, is safety, tolerability, and change from baseline in dystrophin levels as measured by Western blot. Secondary endpoints in this study include measures of muscular function like NSAA, time function tests, and exon skipping. We also have wearables in this study to allow for the collection of data such as stride velocity. There are 3 stages or phases or portions of the DELIVER trial. There's a 24-week multiple ascending dose or MAD portion, a 24-week open label extension portion, and a 96-week long-term extension portion. And so where are we today? I think many of you may have been familiar or are familiar with some data readouts earlier this month. I'm going to be going over high-level summaries, but there are other resources on Dyne's website. We will most likely be doing a webinar with Defeat Duchenne where we can go into elements of the data a little bit more in depth. When we think about safety and tolerability, the safety profile has been favorable for Dyne 251 based on the 54 participants enrolled thus far, with approximately 675 doses administered to date. And when we say to date, the data cutoff was the end of August. The majority of treatment emergent adverse events were mild or moderate. There have been 3 serious treatment emergent adverse events potentially related to study drug in 2 participants in the 40 milligram per kilogram study group. Both participants are home, they've recovered, they are well. Other than the 2 participants with these serious treatment emergent adverse events in the 40 mg per kg Q4 weekly study group every 4 weeks, no participants have demonstrated persistent related anemia or thrombocytopenia or demonstrated kidney injury. And additionally, no participants have demonstrated clinically meaningful changes in electrolytes, including magnesium. All participants at this point are being dosed at 20 milligrams per kilogram, including participants who've been dose escalated following the placebo-controlled period from their starting doses that may have been lower than 20 mg per kg. And then the participants initiated at 40 mg per kg are now being dosed at 20 mg per kg, following that safety evaluation at 40 mg per kg. So I think this is probably the more exciting data and more relevant to the community is what have we demonstrated in terms of our goal of restoring dystrophin as well as function. The patients treated with 20 mg per kg Q4 weekly, so once every 4 weeks, had a mean absolute dystrophin level of 3.7% of normal at 6 months, which we think is remarkable. And when adjusting for muscle content, the Dyne 251 treated group reached 8.2% mean absolute dystrophin. In terms of function, and I think this is what is meaningful for the community, is that improvements were observed in multiple functional endpoints in the 20 mg per kg Dyne 251 group at 6 months, including the North Star, the NSAA, the stride velocity 95th percentile, the 10-meter walk run time, and time to rise from floor. We've also looked at, what is the continued benefit, and continued benefit was seen in these multiple functional endpoints in the 10 mg per kg group, who are now out from 6 months to 12 months. Meaning, I'm going to focus a little bit on the SV95C, in that meaningful improvement was seen in this in both the 10 mg per kg and 20 mg per kg study groups following Q4 dosing. This is a digital objective outcome measure of ambulatory performance in patients' normal daily environment and is approved as a primary endpoint for Duchenne clinical trials in Europe. So very interesting data coming out of this study that will certainly steer us in the future for how our later studies look like. In terms of next steps, DELIVER is a phase 1/2 global trial designed to be potentially registrational. So we have completed our dose optimization, and we will be initiating our registrational cohorts in DELIVER, and we do plan to provide an update on our path to registration by the end of this year. So thank you very much for listening. And if folks have questions, they should always feel free to reach out to me directly.